back to Mini Medical School.
Click and clack.
Click and clack, the Tap It girls. Welcome back. Today you are mini medical students, and when you finish mini medical school, you have to decide if you want to go into a mini specialty. All right? Now, do you guys know what a specialty is? Some of you do and some of you don't. So if you wanted to sort of narrow down your field of medicine that you're going to practice, you could choose a body system, a body organ that you're interested in, like the heart. I'd be a cardiologist, that's a specialty. Or I could be a surgeon, that's a specialty. Dr. Chen is OB, that's a specialty. I'm a gastroenterologist, and that's my specialty. And so tonight, I'm very proud to say that we're going to be focusing on my specialty, me. Let's talk about me. And what's your gut feeling is the topic tonight. And I just wanted to say one little thing. Now, I know that people think in GI, which is my specialty, they think GI is all about stool and poo, and how could you possibly go into a specialty like that? Except for my kids who thought it was cool. So here's the thing about it, honestly. It isn't about the poo, except for tonight's talks. What it really is about is it's about the people. The people in GI are the funniest people in medicine. Am I right? It's true, isn't it?
And that's what you think about as you're doing the scope.
That's right. So tonight, if you decide your mini specialty should be GI, then I want you to lay the gauntlet down and come on into your fellowship, your mini fellowship after you finish mini medical school.
It just says it all has to do with what your gut is feeling and what you can handle. There is no way that I could handle GI, but all of the splashes of fluids that come out of that vagina and that baby is totally fine with me. So, Christopher Damman, acting instructor in GI, UW School of Medicine, a clinical researcher at the Fred Hutch. Current research investigates the role of GI bacteria and how they play in inflammatory bowel disease. And he takes care of patients at the Seattle Cancer Care Alliance with GI diseases. He believes that an imbalance in the colonic bacteria is likely to play a role in inflammatory bowel disease. And he's looking at the potential of therapeutic role of fecal microbiota. We started to talk about that last week, and you guys were really into it, like you didn't want the evening to stop. It was very interesting. No. And these are really nice pictures. See, I like these pictures of babies and stuff, not the colon. Totally unrecognizable to me. His hope is to develop a bacterial-based diagnostics and treatments that have the power to predict and treat GI diseases. He earned his medical degree at Columbia, New York, and he did his residency in internal medicine and fellowship in GI at the University of Washington. We actually kind of have this thing about bringing people from the east to the west, and then they kind of stay, which is kind of nice. This lady, well, what can I say about her? She is the Walters Endowed Chair, Professor of Medicine in GI at School of Medicine, an adjunct professor in pathology, and she's the founder and director of the Pancreatic Cancer Surveillance Program at UW. She takes care of patients in the UW's Digestive Disease Center, and she diagnoses and treats illnesses of the GI tract. And her emphasis involves the origins of pancreatic cancer, which really actually is the thing. I know the lady who discovered the gene for pancreatic cancer. I tell my kids that all the time. And they go, what's the pancreas? So she and I earned our degree at the University of Washington, and she did her internal medicine residency at UCLA. And then came back home. So on that note, you guys are going to spend tonight on the human microbiome.
Well, thank you very much for that very nice introduction. It's a real pleasure to be here tonight and talk about a topic that I'm very passionate about. I think many of my colleagues will attest to the fact that I probably use microbiome every other word in sentences. So I'm going to talk to you about the human microbiome tonight. And just to give you a brief overview of what we're covering, I'll first define what the microbiome is. How does it develop in the human body? How do we study it? What is its involvement in disease? What is its involvement in health? And then some microbiome-targeted therapies. So first, what is the microbiome? Well, to understand what the microbiome is, we have to delve back in history a little bit. This is a picture of Antony von Leeuwenhoek. He was a tinkerer of sorts, and he perfected the microscope back in his day and was able to see microorganisms for the very first time, and some would label him the father of microbiology. He called them animalcules. And here he writes, "I then most always saw that with wonder that, and this said matter, there were many very little living animalcules, very prettily moving." And this is one of the very first descriptions that we have of bacteria. Well, it wasn't until a little bit later, in the 1800s, that the germ theory of disease was developed by Louis Pasteur and Robert Koch, names that you may be familiar with. Pasteurization comes from Louis Pasteur, sterilizing milk and other fluids for preservation. Robert Koch is a name from which we get Koch's postulates, which is a process by which infectious diseases are characterized. And then it wasn't until much later that we began to think of germs not just in terms of disease, but also their role in health, not just bad bacteria, but good bacteria, and understanding the very complex ecosystem that bacteria exist within our body and on our body. And this gentleman here, Joshua Lederberg, was the very first to coin the term microbiome. Now, microbiome is often distinguished from microbiota, but not by everyone. So microbiome, some people use to refer to the collective genetic material that the microbiota contains, whereas the microbiota are the organisms themselves. Well, for the purpose of this talk, I'll use them interchangeably, and that was actually the original intent of microbiome. It was supposed to be the organisms. So let's get a little perspective on the human microbiome. Now, this little dot here represents the weight of the human microbiome. It's on average anywhere between maybe 1 to 4 pounds. This is the average weight of a human, and so it's about 1/100th of a human, and that's more or less what you would expect. Well, let's instead look at the microbiome cells. Now, bacterial cells are very small, so I've represented the microbiome cells as a larger circle. Well, human cells are actually only 1/10 of human microbiome. Pretty amazing. Well, let's take it a step further. How about the microbiome genes? Only 1/100th. So in fact, we're about 99% human by weight, 10% human by cells, and only 1% human by genes. It's a good reason to study the microbiome. Well, I have a little other experiment here. So for reference, here are some things that we can see with the naked eye. There's a coffee bean, there's a grain of rice, and as we delve down into the world of the microbiome, we pass by our first living cell, Amoeba proteus. That's actually a very large cell. We see paramecium, human egg, sperm, and at this point, we see our first bacterium, E. coli. It's 3 by about 0.6 micrometers. And that's about 1/10 the size of an average human cell. We scroll down even further and we begin to see viruses, cellular components, ribosomes, and then you get to the carbon atom. So that gives you a sense of just how small the bacteria are relative to human cells. Now let's look the other way around. Instead of getting very small, let's bring the bacteria up to the size that we can understand how big they are. And we'll start with 1 gram of stool as represented by this Hershey kiss. In fact, there are 100 billion bacteria within a single gram of stool, which I've represented by these little bacteria. We'll increase that to the size of a penny. Well, that's 1 million pennies. That's 1 billion pennies. That's 100 billion pennies. That's how big that stool would be. All right, so we've had some fun with discussions of scale, but what really is the microbiome? Let's delve into this in a little bit more detail and look at the composition of the microbiome. Well, the microbiome contains all three divisions of life, and from high school biology, you may remember there's bacteria, and then there's eukaryota, and that's basically mostly every other cell. Animal cells, plant cells, protist cells. Maybe since some of you have been to high school biology, there's a new division of life which has been understood since we've started sequencing genes called archaea, which is perhaps one of the most ancient divisions of life. And they actually look a lot like bacteria, and it wasn't until we were able to characterize them on a genetic level that we realized that they were quite distinct from bacteria. So, what is a domain? Well, a domain is the highest category of life. You may remember kingdom, phylum, class, order, family, genus, species. It's at the very top. And so there's bacteria, as I mentioned, eukaryota, archaea. Let's focus on bacteria. This is where most of the work has been done in the human microbiome. And we have to delve down a little bit further into the phylum level. Well, at the phylum level, we have all these different groupings. You have Firmicutes, Bacteroidetes, and you can see the others down there. What are these different groupings? Well, I wanted to give you a sense of which bacteria belong to these groupings, why we call them Firmicutes or Bacteroidetes. These are names that are actually derived from Latin. Firmicutes, firm, referring to strong. And cuties, not what you might think, although it's a bit appropriate for a bacteria, but referring to skin, so strong skin. So these are bacteria that are very strong skins. They're gram-positive bacteria. They have very thick cell walls. This is a scanning electron micrograph of strep. Okay, so what bacteria belong to Firmicutes? Well, here you'll see a picture of a skin abscess, and many skin abscesses are caused by staph. You may be familiar with MRSA, which is methicillin-resistant Staphylococcus aureus. But there are also bacteria within this class that are beneficial. So Culturelle, this is an example of a probiotic containing lactobacillus and used to treat pediatric gastrointestinal diseases. Another bad bacteria, Clostridium difficile. This is a bit of a pandemic in hospitals. It's an organism that is very easily transferred from one person to the next, and it requires hand washing with soap and not just disinfectants in order to kill the bug. There are other bacteria very similar to Clostridium that are actually protective to the colon that produce factors that are responsible for the little cells in the colon for growing, butyrate and other what are called short-chain fatty acids. All right, well, let's move on to one of the other phyla, Bacteroidetes. What does that mean in Latin? Well, it's actually bacteroid, bacteria, edes, little. These are little bacteria. These bacteria tend to be some of the smaller ones, smaller than the usual bacteria that you think of like E. coli, maybe up to 1/5 to 1/10 the size. This is a scanning electron micrograph of Bacteroides. And Bacteroides, they're often found in abscesses that are found within the body. So this is a picture of an intra-abdominal abscess. But again, Bacteroides, they can be beneficial. And this is here to demonstrate that Bacteroides may be responsible for harvesting energy from molecules that we ingest, but we're not able to break down ourselves. And so perhaps up to 30% of the energy that we're able to harvest from our food is basically due to bacteria. Some more pathogenic bacteria that fall within Bacteroidetes, Prevotella and Porphyromonas, involved in periodontal disease. Okay, so how about the next one? Actinobacteria. This is a scanning electron micrograph of one actinobacteria, and they're involved again in disease. So this is an example of Propionibacterium acne, responsible in part for the development of acne. This is a beneficial bug within Actinobacteria, Bifidobacterium infantis, another probiotic used in IBS. And moving on in phylum, Proteobacteria, scanning electron micrograph of Salmonella, proteo meaning first bacteria. Involved in traveler's diarrhea, often caused by Campylobacter. and foodborne illnesses, E. coli 0157H7. But again, there are actually protective bugs within this class. E. coli Nisle 1917 was isolated from a German soldier in 1917, World War I. And this is used as a treatment for IBS, or actually IBD. And H. pylori, the organism that causes stomach ulcers and gastritis. This is also a proteobacteria. All right, another phylum, Fusobacteria and Cyanobacteria. All right, so beyond bacteria, let's delve into the realm of eukaryota. So these are non-bacterial cells, and this is a scanning electron micrograph of yeast involved in yeast infections. So here you can see infection in the mouth of yeast, but also protective. So this is Saccharomyces boulardii, which is used as adjunctive treatments in antibiotic-resistant Clostridium difficile infections. This is a Giardia lamblia, scanning electron micrograph of Giardia, also eukaryote. A worm, intestinal worm, Ascaris lumbricoides. But actually, another worm is used as a treatment for IBD, primarily in Europe, Trichuris trichuria, otherwise known as the whipworm. All right, and actually, now we're up to Archaea, one of my favorite divisions. This is a scanning electron micrograph of Methanobacter smithii. Now, we don't find these particular bugs in our guts, but one of the amazing things about Archaea is they live in some of the most extreme environments in the world. And this is a picture of a thermal spring in Yellowstone. And the very beautiful colors, the sort of prismatic colors that you see around the outside, are actually being produced by bacteria. And another so-called extremophile, because they can live in very extreme environments, are these halophiles, organisms that like salt. And the pink that you see in this salt lake is produced by the bacteria and is used for harvesting energy from the sun. And perhaps the most extreme environment of them all is the cow gut, where we find Methanobacter, responsible for production of methane. In fact, 1/3 of you in the audience have this bacteria in your gut, and 1/3 of you will produce methane. Now, viruses are often forgotten when we discuss the microbiome, but they are very important, and so I include them in this slide here, and probably are involved in disease processes as well as health. This is a scanning electron micrograph of a bacteriophage. These are bacteria that viruses that infect bacteria. Now, how do we develop, or actually, how is the microbiome different across our body? And in fact, it is quite different. So here you see pie charts, and the colors within the pie charts represent the different phylum that we've discussed so far. So Bacteroides, Firmicutes, etc. Now, on the left here, you'll see the blue, and that's actually actinobacteria, and that's pointing to most of the external surfaces of the body, and the external surfaces are actually dominated by that. The red are the Bacteroides, and the internal surfaces of our body are actually dominated by those. And amazingly, if you look across even the external surfaces of the body, the right hand versus the left hand, the knees versus the elbows, you'll find different microbiota. And within the gut, as you traverse from the upper gut down to the lower gut, you have different microbiota. The upper gut bugs tend to like oxygen more, and the lower gut bugs tend to like oxygen less. And in fact, the concentration of the bugs increases exponentially as you get down to the lower parts, where our stool contains 100 billion bacteria per milliliter. So how do we develop this amazing microbiota? Some actually call it another organ. And how does it change? Well, for the most part, we're sterile at birth, but in fact, we're not entirely sterile. And there are some bugs that are found in meconium, the first stool that's passed by babies. And as we develop, our environment influences dramatically the microbiota. And so formula-fed babies versus breastfed babies have different microbiota. And as soon as we start eating solid food, the microbiota starts to take on more of an adult characteristic, and by the age of 1, it's more or less mature. Various things influence the microbiota through development, so antibiotics and nutrition, age, obesity. And in fact, as we get older, the microbiota changes as well with different proportions. All right, so I've said a lot about the microbiota, but I haven't said how we know all of this. So how do we study the microbiota? What tools do we have? Well, traditionally, and back to the days of Koch and Pasteur, we had bacterial culture, and that was pretty much it. Culture where we would isolate single species of bacteria and understand them in sort of a reductionist approach. But as you might conjecture, it's a really complex mixture of bacteria. And the bacteria are all sort of working in concert. And so perhaps it's better to understand the bacteria in that context in some cases rather than isolating them. Now, one tool that's developed very recently, being within the last 20, 50 years, is gene sequencing. Here you see next generation sequencers, Illumina, Ion Torrent, Pacific Bio. And what these sequencers allow you to do is basically profile the specific bugs that are present within the microbiota. That's using one approach, and that's a targeted approach. And so we use a very specific gene that's present in all bacteria, but has very slight variations within it. And you can imagine those variations a bit like a barcode. And so we can sequence that specific gene and compare that to a database and see exactly what bugs are there and know what proportion they're there. And we get an output very like this. It's a heat plot. And so the bars that you see here, the darker they are, the more bacteria you have for that specific species. Species across the left, and then individual samples across the top. But there's another way of sequencing the human microbiome, and that is using a metagenomic approach. And rather than targeting a specific area within every bug, we sequence everything. And then we can develop these very, very complex metabolic pathways based on the genes that we find present. So, all of this sequencing, what has it amounted to? Well, there have been two major projects, the Human Microbiome Project and MetaHit. Human Microbiome Project is an NIH-sponsored project here in the US. It was initiated in 2008. It's actually now complete, and it cost $115 million. Now, for comparison, the Human Genome Project costs on the order of $4 billion. So you can imagine just how much cheaper the sequencing has gotten. There's another initiative, the MetaHit project. It's a European cooperative. That was also started in 2008, and that cost 11.4 million pounds. But ultimately, we need to come back to bacterial culture because these studies will allow us to make correlations and will give us a hint at maybe what these bacteria are doing. But ultimately, we need to test these hypotheses. And so for that purpose, we need to be able to culture these bacteria, maybe 50% by the best estimates or the highest estimates of which we can currently culture. So new techniques are necessary in order to be able to do that. And if we can culture these bacteria, we can study them using these model systems. So we can grow human cells in tissue culture and add the bacteria to these cells and see how the cells react. Are the cells inflamed or are they happy? And we can look at the molecules that they produce as a readout for that. And we can use animal models as well. And in fact, germ-free facilities are a very powerful tool that we have for studying the microbiome. Germ-free facilities you can think of like a mouse in a bubble. It's a mouse that's never seen any bacteria except for the bacteria that you give to it. These are called notobiotic facilities because they're known, noto, bios, bacteria. So you can study the bacteria of choice that you're interested in a very defined environment. So what have we learned using these systems about microbiota and health, and ultimately about microbiota and disease? And what new therapies can come of this? Well, in health, the microbiota is in balance. In other words, it's in eubiosis. And so on the left here, you see the symbionts. Those are the good bacteria. And on the right, you see pathobionts. Those are bad bacteria. In the middle are commensals. Those are bacteria that are neither here nor there. Now, microbiota and health, they serve many different functions. So there's metabolic functions, there's protective functions, there's structural functions. And let's examine each of these in a little more detail. So metabolic functions, energy metabolism. I alluded this to you before. The bacteria help us digest the food that we eat. And in fact, animals that are reared in a germ-free environment that don't have any bacteria require 30% more calories in order to maintain the same body weights as their conventional counterparts. The microbiota are responsible for production of vitamins, most notably vitamin K. And folks that may be on warfarin, which is a blood thinner that's used for people that have had heart attacks or strokes, will know that if they take antibiotics, their INR level, which is a measure of how thin their blood is, which is what the warfarin does, is thin the blood, will be dramatically affected. And one mechanism by which that happens is the bacteria that produce vitamin K are essentially killed by the antibiotics. Bacteria, microbiota, also are involved in drug metabolism. So it's not just your liver that breaks down the drugs that we ingest, but even before they get absorbed into the body, the microbiota starts breaking them down either into inactive metabolites or actually the active compounds. And these are some examples here. The microbiota is involved in development of the gut, and you'll see here, this is a slide of a rat gut. And you can see the thickness on the right side is actually much greater. That's in a conventional mouse versus on the left. And the architecture is distorted on the left. And so the microbiota is involved in development of the architecture of the gut. Now, this is a busy slide. Don't worry about the details. But this is just to say that the microbiota is profoundly influencing our immune system, not just in the gut, but also distal to the gut. And so it plays a role in diseases like potentially even asthma and skin diseases. It protects us from infection. All right, so moving on, microbiome and disease. So this is a slide of dysbiosis. In other words, bacteria that are out of balance. And so you see on the right side, the bad bacteria, the pathobionts outnumber the good bacteria. And there are many different things that contribute to dysbiosis. So our host genetics, we can just have a predisposition to having dysbiosis. Various lifestyle factors like diet and stress, early experiences that we have in hospital environments, and various medical practices, antibiotics, vaccinations, hygiene. So dysbiosis and disease, as I mentioned, the microbiota is involved in just about every organ system in the body. So being a gastroenterologist, I'll focus a little bit more on the GI diseases. So IBS, irritable bowel syndrome, it's a disease characterized by either diarrhea or constipation and pain in the abdomen. It's profoundly influenced by the microbiota, and there are a lot of exciting studies showing a dysbiosis in IBS, and a lot of the therapies that we have, which I'll talk about in just a minute, impact the microbiota. Now, IBD, often confused with IBS, but a very different disease. It stands for inflammatory bowel disease. In this disease, you actually see inflammation in the gut, which you don't see in IBS. And again, there is dysbiosis that's found when we sequence the human microbiome from patients that have IBD. Interestingly, colorectal cancer, there have been a number of recent studies showing associations between specific species in the gut and colorectal cancer. Obesity. Amazingly, there's a connection between the brain and the gut, and it goes in two directions. And so the bacteria can influence our states of anxiety. That's seen in model organisms like mice, and vice versa. Our brain can influence which bacteria are present in the gut based on how anxious we are. It can change how leaky the gut is and how much inflammation might be present in the gut. So an example of the gut-brain axis in GI is hepatic encephalopathy. Now, this is a condition that's seen in patients that have cirrhosis of the liver. And in cirrhosis of the liver doesn't filter out from the blood toxins that are produced by bacteria. And those toxins go to the brain and they cause confusion. And there are many other diseases as well. There's heart disease, autoimmune disease, allergy and asthma, arthritis, dermatitis, burns, and many, many others that we probably don't even realize. So that's all fine and good, but what's the bottom line? In medicine, we like to study things because we can develop new therapies. And so what are some of the new therapies that all of this new research has developed? Well, antibiotics, as one might imagine, specific antibiotics that target the bad bugs could potentially be good and bring things back into eubiosis. But also, we can use good bugs like probiotics and prebiotics, which are substances like lactulose that are given in hepatic encephalopathy that promotes the growth of good bacteria and that are often found in combination with probiotics in formulations called symbiotics, bringing things back into eubiosis. So here are some real-world examples. Now, antibiotics for IBS. There's an antibiotic called rifaximin, and it doesn't get absorbed into the body. It's only present in the gut. And it's been shown to be an effective therapy for certain subtypes or certain patients with IBS in a placebo-controlled trial. Now, very recently, I believe it was either last month or the month before, in the journal Gastroenterology, there was a landmark study showing that a specific diet called the FODMAPs diet is effective for irritable bowel syndrome. FODMAP standing for fermentable oligosaccharides, disaccharides, monosaccharides, and polyols. And these are basically, for the most part, different types of carbohydrates that bacteria will have a heyday with and produce lots of gas. And it's perhaps patients that have more gas associated with their IBS that respond best to this type of treatment. Now, this is a lot more controversial, and traditionally, the teaching is that diet does not affect inflammatory bowel disease like Crohn's disease and ulcerative colitis. But there's very recent literature coming out from the pediatric literature showing that maybe that's not entirely the case. There are at least 2 new studies showing that a specific carbohydrate diet is actually efficacious for pediatric Crohn's disease, not just in decreasing symptoms, but decreasing inflammation seen in the gut. I spoke about hepatic encephalopathy. Well, lactulose is an example of a prebiotic which good bacteria like. They use it as a substrate for growing. And so this will change the balance, get rid of the bacteria that are producing the toxins that don't get filtered out by the liver and that go to the brain and cause confusion. And then certainly probiotics. And there are lots of probiotics. You can go to Whole Foods and probably have a choice of 10 to 50 different probiotics. And some have been studied in a little more detail, and here I've highlighted 4 of them: Culturelle, Align, Florastor, and VSL number 3. Culturelle tends to be used more in pediatric viral gastroenteritis. Align, it's a bifidobacterium formulation, and it's been studied primarily in IBS. Florastor, this is Saccharomyces boulardii, and this is a treatment that's been validated as an adjunct for Clostridium difficile colitis. And then VSL number 3, which is a combination of 8 different probiotics, including lactobacillus and bifidobacterium. And this is used for IBD, inflammatory bowel disease. And then lastly, not to steal the thunder of Dr. Brentnall, but to just very briefly mention fecal transplantation, which some might say is the ultimate probiotic, giving bacteria that have been adapted to the gut back to individuals who have certain bacteria depleted. Very well documented as a treatment for Clostridium difficile and being actively investigated for other diseases like ulcerative colitis, which is my area of interest. So what have we discussed? We've discussed what the microbiome is, and it's the combination of pathogenic and neutral and protective microbes that live on and within us. And how does it develop? Well, in early development, we get it primarily from our moms. Our diet influences, antibiotics, nutrition, health, and age influence it. How do we study it? Traditionally culture, more recently sequencing, But in fact, we'll need to go back to culture and tissue culture and animal models. How is it involved in disease? Well, it helps us harvest energy from our food. It's involved in vitamin production, drug metabolism, helps our gut develop, involved in immunity and protection from infection. In disease, IBS, IBD, colorectal cancer, obesity, hepatic encephalopathy, and many others. And therapies, antibiotics, probiotics, prebiotics, symbiotics, diet, and fecal transplantation. That's it.
That was great. That was fabulous, really fabulous. Okay, so that was to give you sort of a background overview of why poop is important. And so I'm going to talk about why poop is deadly. And so I'm going to talk about a deadly diarrhea that has become an epidemic in the United States and also in Europe. And the salvation of the stool, how your stool can get you in trouble and then stool can get you out of trouble. So over the last decade, even the New York Times has noted that there has been an epidemic of this deadly infection, a type of bacteria that causes terrible diarrhea. And in the United States, it's the most common cause of deadly diarrhea. If you were in Africa, we have other causes of deadly diarrhea, but here in the United States and in Europe, it's C. difficile. And what happens is this Clostridium difficile is a bacteria which actually Chris alluded to. And what happens is when you get treated with antibiotics, your gut microbiome gets out of whack and it kills off a lot of the bacteria, and what grows up can be this bacteria, the bad bacteria called C. difficile or C. diff for short. Now this bacteria has little spores that it makes, and that's how it propagates itself. And these spores are like little BBs. They're tiny little hard little items that are really hard to kill. And so they can hang around, these spores do. And it's become a menace in hospitals, nursing homes. So what happens is the spores sort of hang around, and they could be on any surface, actually any surface anywhere. And so you have to be very careful to get rid of these spores. And people that have been treated with antibiotics, your gut microbiome is out of whack. All you need to do, touch a surface, lick your hand, which I know a lot of you do. Especially after you eat your sandwich, you lick your hand, go like this. So that's how we pick up those spores, and then we get infected. So when we get this infection with this bacteria, it causes huge inflammation in the colon, which is the large intestine. And when you have big inflammation in there, we call that colitis. And so, interestingly, C. diff, our bad organism, is actually naturally found in a lot of people's guts. But the other ways that we can pick it up, if it doesn't happen to be in your gut, is that you can find it on the surface of things. And it's stool contamination, where someone might use the bathroom and then they come out and they didn't wash their hands, and then they push the elevator buttons, which is why I use a pencil to push the elevator buttons. So we talked about the gut bacteria becomes sort of unbalanced after antibiotics, and that sets up an ideal situation for these C. diff bacteria to grow, and they make toxins, poison. They make poison. And so as you saw earlier from the slides that Chris Damman showed you, there's a wide variety of bacteria, and it gets even more numerous in terms of the species as you go down the colon. Normally you should have a wide variety in there, but after you get antibiotics, you get rid of many of those bacteria, and what grows out might be this Clostridium C. difficile. And when you do a colonoscopy, it's really interesting. It's like I know right away. I can tell my trainees, you know C. diff when you see it. It looks like someone took scrambled eggs and threw them onto the lining of the colon. So all this yellow stuff that you're looking at, which looks like scrambled eggs, are actually giant pus balls caused by the bacteria. It's kind of gross, but we don't have to touch it, so don't worry about it. So when you look under a microscope, this is what you would see. It would be colorful and it would not smell bad. The problem with C. diff is that over the last 5 to 10 years, it's become more drug resistant and more deadly. And so there's actually a strain out that is really highly lethal, and it's getting around, and we're against that. As a result of that, hospitalization for C. diff has really skyrocketed since the early 90s, when about 85,000 US individuals were hospitalized. This chart only goes to 2010, but in 2013, 500,000 people hospitalized for C. diff. So that's a lot of people coming in with diarrhea. So let's talk about the epidemiology. That's a little bit about how this all comes into action. So it commonly occurs in patients that are in a hospital or in a nursing home. Not always, but it can occur there, and the reason for that is people with C. diff go to hospitals and then they push the elevator buttons. You know what I'm saying? A lot of you are going to buy a pencil tonight, aren't you? Yeah, but it's just not the elevator buttons, it's more than that. They touch the stairwell and the wall and the light switch, and they go in the bathroom and splash things around. It's just the nature of the thing. And then the same thing happens in nursing homes. So it's just flying around here. As if that was not enough. The spores, which are like the little baby C. diffs waiting to be born, they can survive cold and hot and dry and wet. It doesn't matter. You throw it out there and you could make it freezing and then come back a month later and that spore is right there waiting for you. Has your name on it. So actually, your basic, like if you say, well, I'm gonna use some Lysol on that. No. Sorry, Lysol doesn't work. What about, I'm gonna use a green product. I'm gonna use a green product made out of orange oil and tea tree stuff. Sorry, still there. So really, the way to get rid of this is bleach. You have to get a big box of Clorox and just throw it around. And you know, it's interesting because for a long time there, I think part of the epidemic was people don't throw around bleach like they used to. And so it became a problem until we really realized it's bleach or nothing, really. That's kind of what we're all about. Now, stool is a funny topic. I mean, let's face that, poop is funny, but death by poop is not funny. So 14,000 people are listed by their death certificate of dying of C. diff last year. But that number is actually probably not accurate because that is, if you look at the death certificate and the top word says cause of death, C. diff, it gets listed. But if it's not on the top line, it's not listed. I'm going to show you how that works. It's probably closer to 30,000 people died from C. diff last year. That is the same number of people that died from random accidents. Here's a birth certificate. Look, cardiopulmonary arrest. That means their heart stopped and their lungs stopped. Sepsis and dehydration is another thing listed on there, and the third thing listed on there is C. diff. Well, guess what? C. diff caused the sepsis and the dehydration, and then their heart stopped. So that one didn't get counted, but you know that guy died of C. diff colitis. So this is an example where the death certificate is not really truly counting the number of people who have died of this disease. Okay, so let's talk about treatment. So normally what we do is something that Chris Damman was talking about. When you have a bad bacteria that gets in there, we can throw antibiotics at it to kill the C. diff. So we've got 2 really good or 3 really good antibiotics that we use. And it works pretty well, except for about 20% of patients, which is not a small number. 20% of patients relapse. The C. diff comes back. And so we go at them again with more antibiotics, and 50% of the time, it doesn't work. So once you start relapsing, you're really prone to relapsing, and the more you relapse, the more you relapse. You know what I'm saying? You guys tracking with me here? Some of you are sleeping. All right, so what do we do for those people? Fecal transplantation for gut repopulation. Repopulation. All right, Scientific American, this is a scholarly journal. So what we do is, we know that even if we keep throwing antibiotics at you, really your gut microbiome is out of whack. It's out of whack, and even if we keep throwing antibiotics at you, and maybe even some probiotics, it's out of whack, and we can't get it back in whack. So what we need to do is take some stool from a really good-looking person and put it in you, and then you're going to be fine. So how do we actually do that? Well, first of all, you have to find a donor. We actually call the person who is going to give the stool away the donor. And so usually that donor is somebody that you love, but let's say you don't have anybody like that. Then it would be somebody you're related to, like a spouse. Or you could fill in a blank, maybe a friend. You know, and I always thought that, like, if I wanted to make money and get out of GI, I would get Brad Pitt's poo, and then I could sell that, because what would be better than movie star poo? So what we do is we get a stool sample, and it actually looks like that. And we send their poo off to be tested to make sure there's nothing ugly in there. And if there's nothing ugly, it's a go. We say it's a go. And so we get the stool. I won't delve into how that happens, but use your imagination. And we put it in a blender. Okay, it's not like it's just any blender. It's the poo blender. It's in the GI suite, but we do just buy it at, like, Costco or, you know, Kmart. It's not like it's a special blender. It's your basic. You could use it for margaritas if you weren't using it to scramble up the poo. So we whip up the poo, and then we put it through a filter so the big chunks don't go, because that's ugly. So we get the solvent, you know, kind of liquidy stuff, and we put it, and we wear gloves. Like, actually, I wear a body bag when I'm doing this. So you put it in these little tubes, and then we do a colonoscopy, and this is actually how a colonoscopy is done. Nobody actually holds the scope. We run out of the room after the patient's asleep. Good luck with that scope, but I hope it finds its way up the colon. No, actually, that's not true. We actually push it up the colon. And then we take a syringe, and we have ways to push the poo down the scope in a very clean setting. Doesn't even smell bad very much. And we put it into the patient who is asleep, the patient who feels nothing, nothing. They are snoozing through the whole thing. Easy peasy. The beauty of it is that it works great. It doesn't work a little, it works great. 95% of the time, it works. It is amazing. You take people who are sick, and you do a fecal transplant, and they feel fantastic. They are cured because they got somebody else's poo. Okay, so let's talk about fecal transplantation and the power of poop. First of all, in the beginning, this was like, people would go like, really? What? What you say? You know, like, what? You know, even the GI doctors was like, I'm not doing that. But because it works, we tend to do things in medicine that work. We're kind of for that, and it works really great. So now it's being done all across the nation. Now, the physician who actually does this has to be kind of FDA approved. You have to call the FDA and register in with them that I'm going to be passing the poo, and they have to like okay you to be the person that does that. So it's just not any willy-nilly person that can be doing this. It has to be someone who knows how to dial up the FDA. Now, a lot of people want to do this, but they don't want to have the colonoscopy. And I say, why? Why not have a colonoscopy? We give great drugs. You sleep right through it. It's fantastic. But a lot of people are against the colonoscopy part, so they'd be like, you know, I'd rather eat a lot of pills. Now, they're called stool pills. Personally, that wouldn't be my choice, but there are people that that's their choice. So what they do is they sort of condense down the poo, take out all the water, and they stick it in little gelatin capsules, and you have to eat 30 of those capsules. Big bag like this, and they're brown, just saying. Okay, but it actually works. Saves lives. What could be better than saving lives? From an epidemic. I'm saying this is great stuff. Now, is the only answer is to transplant stool for everybody? No, the thing we really need to do is get on top of prevention of C. diff colitis. Now, besides using a pencil to push the elevator buttons, we have some other methods for doing this. So first of all, don't just eat antibiotics because they're there. I know a lot of you do that. You're like, I got a little cough. I'm going to see my doctor and get some, you know, amoxicillin. Really, it would have gone away without the amoxicillin. So unless you really need antibiotics, probably don't take them. Also, we know probiotics can be helpful. So just throw some of those in. If you happen to be on antibiotics and then you take some probiotics, you'll get that gut flora back kind of in the happy zone. Hand washing. I know that your mother said you had to wash your hands, but she was right. You got to wash your hands, and you got to do it really good and use some soap. Sort of scrub, 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 scrub. It actually gets things clean. Now, here's another tip. If you get diarrhea after you use antibiotics, so some people get diarrhea while they're using antibiotics, and that is related to the antibiotic itself. But if you finish a course of antibiotics, you're fine, fine, fine. Now I have terrible diarrhea. You should contact your doctor because you might have C. diff. And sometimes people don't know to do that, and those are the ones that can get really sick. So we want to nip it in the bud in the beginning. In the hospital, the hand washing programs that we use, because we don't advocate using bleach on our hands, we do use a particular kind of soap that's called chlorhexidine, and it actually does kill C. diff. So all these years, people have been using a lot of alcohol-based soaps, you know, Purell and like that. But if it doesn't have chlorhexidine in it, it won't get rid of the C. diff. This also changed the way that we clean the rooms after a patient leaves. We have to throw around a big bucket of bleach. You know, none of this, I swept the bed and we're ready for the next person. We really have to deep clean to get rid of those spores. So I'm going to close and say to you, what can brown do for you? Okay, thanks.